At the beginning of this lecture, I want to say that this week I've got from our library the 14th edition of the Longman's Medical Embryology. It was edited in 2020, this year, just the most recent edition, and the 8th edition of the Ross Histology. These are the two textbooks. Unfortunately, they are not available for students, but if you want, you may download from the internet previous editions. I want to say that really we analyze different textbooks, the most recent editions, to concentrate all the information in our lectures for you to complement your basic textbooks. So you don't need to analyze many textbooks, you don't need to search in the internet to get some additional information. We do this for you, and all you have to analyze is the basic textbook and our lectures. So let's get started. And today we are going to discuss female reproductive system. It is very important to understand how cyclic changes occurs in not only female reproductive system but in the whole female organism. And also it is important to understand the hormonal control of those cyclic changes, as it helps us to understand how different drugs and pharmacology works, how we can correct the menstrual cycle, terminate the pregnancy, or prevent the pregnancy, or maybe some tumors, how they are treated. To understand all that treatment strategies, all those treatment strategies, you have to understand how this system works under normal circumstances. So let's start. And in female reproductive system, we first discuss the ovary. So in ovaries, we can find the surface is covered with a simple cuboidal epithelium, which is called germinal epithelium. It was called so because it was thought that it gives rise to oogonia, to the germ cells. But nowadays we understand that really yolk sac is the source of primordial germ cells, but the name was preserved. But keep in mind that it doesn't give rise to germinal cells. Germinal epithelium is well known because it gives rise to ovarian cancer. So if you've ever heard, the majority of cases derives exactly from this epithelium, because during the ovulation, the epithelium ruptures, and those frequently occurring damage once in 28 days during the reproductive period seems to affect the epithelium negatively and make it prone to the malignization. And then, after the germinal epithelium, under this epithelium, we can find the medulla and the cortex. In the medulla, loose connective tissue contains blood vessels, nerve endings, while in the cortex, we can find different stages of follicles. So follicles on different steps of their maturation, and also the yellow bodies and corpus luteum. And that's exactly what we are going to discuss. So in the structure of the ovary, we pay attention to different structures located in the cortex. And we start from primordial follicles. They are the most immature, and they are present since the birth, and they represent the primary oocyte, which is in the center, and squamous follicular cells. They are flattened, and there is only one row of them. You remember follicle cells, they accompany oocyte and assist, and they are used as a servants to produce some nutrients, to take out wastes of the oocyte, so they are assistant cells. In the oocyte itself, several structures should be mentioned, well, maybe not very important, but Balbiani body, this is the concentration of almost all organelles. They are grouped together close to the nucleus, and also annulate lamella. These are something like a stack of nuclear envelope profiles with structures resembling the nuclear pores. Then, these are the most numerous follicles. They represent the reserve of oocytes for the whole reproductive period. So, unlike male reproductive system, where primary spermatocytes are generated along the reproductive life, here in female organism, all primary oocytes are already created before the birth, and they are retained here in those follicles. And then Starting from the puberty, menstrual cycles begin, and every menstrual cycle, every 28 days, one of those follicles becomes mature. So along with those 28 days, every menstrual cycle, several follicles, 10 to 20 follicles, they start to grow. So only one ovary works every menstrual cycle, left or right, and they work alternatively, one by one. And out of those 20 follicles, only one become mature, but they start to grow. And after primordial follicles, the next stage is primary follicles. The difference is that in primary follicles, follicle cells, they become cuboidal. So if in the ovarian, in the primordial follicle, the cells are flat and squamous, here they become cuboidal. Then several layers of those cuboidal cells appear, and we see that they bordered from the connective tissue surrounding the follicle with a basal lamina. Then another sign of the primary follicle is the appearance of the zona pellucida. You remember this is the crucial zone which is penetrated by spermatozoa acrosomes. And through the zona pellucida, follicle cells send their processes to the oocyte, and oocyte in turn sends its processes, and they contact with each other via the gap junctions. So they exchange small nutrients and metabolites, and in this way granulosa cells they affect the oocyte. So now we call them granulosa cells because they contain nuclei, and there are lots of their nuclei like granules around the oocyte. So now they are called granulosa cells. Then, as follicle continue to grow, the number of follicle cells increase, and they start to produce liquid. And this liquid creates small cavities inside the follicle, and follicles with cavities we call secondary follicles. And together primary and secondary follicles are called growing follicles as they start to enlarge. And what we have to say is that oocytes inside follicles are retained in the prophase of the first meiosis. So almost all oocytes found in the ovary, they are in the prophase, they are arrested in the prophase of the first meiosis. And to maintain this arrest, several factors work. First of all, follicle cells, they produce cyclic GMP, guanosine monophosphate, and transport via the gap junction this cGMP to the oocyte. And in the oocyte, they inhibit the phosphodiesterase, and in this way they prevent cyclic AMP breakdown. So cyclic adenosine monophosphate increases and its concentration is high, and this is exactly what helps to maintain the meiotic arrest. Another factor inhibiting the meiosis is the C-type natriuretic peptide. This peptide also is produced by granulosa cells, and its concentration is highest in the immature follicles, and it prevents the maturation of the oocyte. So, next, after fluid is produced, small cavities appear in the follicles, and this is what we call the secondary follicle, but eventually cavities they fuse with the formation of one single large cavity, which is called antrum. And such follicles are called now mature, Graafian, or tertiary follicles. And these follicles should be considered in details, what cells they contain and what they produce. In these follicles, we have still primary oocytes surrounded by the zona pellucida and granulosa cells and the antrum. But except granulosa cells, around the follicle in the connective tissue, two layers are formed. They are called in Latin theca interna and theca externa. Theca interna cells, they are represented by typically steroid-producing cells. And if you remember male reproductive system and Leydig cells found between the convoluted tubules between the seminiferous tubules. Here, their homologs are present. These are the theca interna cells, they are steroid-producing cells, and they are also sensitive to luteinizing hormone, as well as Leydig cells. And around them, we can find a layer of smooth muscle cells together with collagen fibers, and we call this layer theca externa. And its function is to contract the ovary during the ovulation. No, I'm sorry, to contract the follicle, yeah, to expel the oocyte during the ovulation. We'll discuss this a little further. And now what we have to discuss in details. This is the interplay and the interaction between the theca interna cells and granulosa cells. And their interaction in the way of estrogen synthesis. It is important to say that neither theca interna nor granulosa cells contain the full set of enzymes to produce estrogens, so they should work together, and different steps of estrogen synthesis are performed in either of them. So let's trace this process. This is very complicated, but you have to comprehend in general. Besides, this photo is taken from the last edition of the Histology, written by Ross, because it is not present in the internet. So, theca interna cells, here they are in purple color, they have receptors to the luteinizing hormone, so they are affected by luteinizing hormone, and it triggers the chain of event with the formation of the cyclic EMP. But except luteinizing hormone, they also expose low-density lipids receptors. Low-density lipids are captured from the bloodstream. You remember those lipids, yeah? They contain cholesterol. And cholesterol is liberated from low-density lipids, and under the action of side chain cleavage enzyme complex, is converted into the precursors of androgens. We do not ask you to remember all those intermediates like pregnenolone, dehydroepiandrosterone, and so on, but in general, you have to comprehend that under the action of luteinizing hormone, cholesterol is converted into androgens in the theca interna cells. At least this. You have to at least remember that theca interna, they produce androgens under the action of luteinizing hormone. Okay, then those androgens in form of testosterone or androstenedione, they are transported to the follicular cells of the granulosa layer, and here, and that's what you have to remember, under the action of P450 aromatase should be remembered for 100%. Aromatase helps to convert androgens into estrogens. Okay, these are the key events occurring here. So collectively, theca interna and granulosa, they produce estrogens, but different steps are performed in different cells. In this way, theca interna cells, they are sensitive to luteinizing hormone. Follicle cells, they are sensitive to follicular stimulating hormone, as they contain special receptors. Okay, so estrogens, they are produced to the follicular fluid. And in general, we can say that follicles, especially mature follicles, are very active source of female sex hormones, particularly of estrogens. But notice, once again, I want to highlight that estrogens, they derive from androgens. So primarily, male sex hormones are produced, and later they are converted into female hormones. Why this diagram is very important? Because, for example, in oncology, to stop and to prevent the growth of hormone-sensitive tumors, sometimes aromatase is blocked. So in different types of therapy, one of the strategies is to block the aromatase. In this way, estrogens are not formed, and tumors sensitive for estrogens, they degenerate. So it is important to remember. And not only this step could be blocked, but also all the others. So you have to know this diagram. Then, not all follicles become mature. Some of them degenerate. As I've said, every menstrual cycle, up to 20 follicles, they start to grow, but eventually only one destined to ovulate. What happens with other? Up to 19 follicles could degenerate, and this is the normal event. And such follicles are called atretic follicles. So they do not become mature, they undergo back development, they do not ovulate. And what happens with them, how they degenerate? First of all, their granulosa cells, they undergo the apoptosis, as we can see here. You may see the normal follicle and nearby the atretic one. You can compare what happens. Here granulosa cells are normal, and here they undergo the apoptosis. The cavity collapses, the oocyte itself undergo apoptosis and also collapse, shrinks and collapses. Then many immunocompetent cells and connective tissue invades the granulosa and the cavity and replaces it. So eventually the atretic follicles, they disappear and they are replaced with the connective tissue. What is the function of those follicles? Only one maturing follicle is not enough source of estrogens. So up to 20 follicles are needed to produce enough amount of estrogens. But only one should ovulate, because in humans, uterus destined to carry only one fetus during the pregnancy. We have only single one cavity, and normal pregnancy is considered when only one fetus is inside. So only one ovum should, or only one follicle should ovulate. Then this factor is believed to inhibit the atretic process. So in atretic follicles, neural apoptosis inhibitory protein is not found, so it prevents the apoptosis in normal follicles. In case of its absence, follicles, they undergo the atretic process. In some animals, follicles undergoing atresia, they do not disappear at once abruptly, but their theca interna cells, they disperse in the ovary and continue to produce androgens. In humans, some cells called Hilar cells are found in the hilum of the ovary. Such cells are homologs of Leydig cells, as it is written here. They contain also Reinke crystalloids. They responsible for the secondary sex signs formation during the puberty, and also they perform important role in menopausal period. They produce androgens, and if they give rise to tumors, then the sign of such tumors is masculinization, so the formation of male signs. Why Hilar cells are put here just after the atretic follicles? Because in some animals during the process of atresia, theca interna cells, they form something like Hilar cells. But in humans, Hilar cells are present, and in very small amount. Maybe they are not so important as in animals, but we should keep in mind and remember about them because of tumors possibly which might arise from these cells. Sometimes androgen producing cells exist, and if in small amount it is normal event. Okay, then after we have discussed primordial follicles with a simple squamous epithelium, primary with a simple cuboidal, secondary with small cavities, and at least eventually metrographian follicle with granulosa, theca interna, theca externa, we should say that eventually one follicle ovulates, and this is the critical event for fertilization, as only when oocyte occurs in the fallopian tube, only there it could be fertilized. So ovulation is the process when secondary oocyte is released out of the ovary. Notice that in ovaries, follicles are insulated from the surface, and if in male reproductive system spermatozoa are formed inside convoluted seminiferous tubules, and tubules are communicated with the retitestis, with the afferent ductules, and up to the urethra. Spermatozoa travel along the continuous system of tubules. In female reproductive system, oocytes, they are insulated in the ovary, and to liberate, to expel the oocyte, the wall of ovary should be ruptured, and that's exactly what occurs during the ovulation. Rupturing of the surface epithelium results in the release of the oocyte, and this oocyte should be captured by the fallopian tube, as again, fallopian tube are not continuous with the ovary. There is the space between them, unlike male reproductive system. So, what controls the ovulation? First of all, this is the luteinizing hormone, and as it is written here, the highest concentration of luteinizing hormone results, together with follicular stimulating hormone, results in the ovulation. What stimulates ovulation? First of all, this is increased volume of the Graafian follicle, because at the end of its existence, this Graafian follicle, it contains more and more fluid. At the same time, granulosa cells, they stop proliferate, but they still produce fluid. And as follicle doesn't grow, but pressure inside increases, as a result, it ruptures. Then another event helpful in the ovulation, this is the proteolytic action of plasminogen. And as I've said, in the theca externa there are some smooth muscle elements, and by their contraction they help to expel the oocyte out under the action of prostaglandins. So once oocyte is released to the fallopian tube, what happens with the remnants of this follicle? They are transformed into so-called corpus luteum. And corpus luteum is translated as body, yellow body. It is of yellow color because of the pigment lipofuscin. And what is it? First of all, corpus luteum is formed predominantly by the granulosa cells. So the cells of granulosa layer, after they have expelled the oocyte, they start to proliferate, and they produce estrogens, progesterone, and inhibin. You remember inhibin inhibits follicular stimulating hormone by the mechanism of the negative feedback loop. Then, not only granulosa cells start to proliferate, but theca interna cells as well. So, such cells are called theca luteal cells. They also produce progesterone and androgens, but they represent the minority of cells in the corpus luteum. So, corpus luteum is a transient endocrine gland producing in general progesterone, but some amount of other hormones as well. And the main function of this yellow body to produce progesterone and to prepare uterus for the pregnancy. And that's what we'll consider later, what happens in the uterus under the action of this hormone. And there are three stages of corpus luteum formation. This is the proliferation and vascularization, when cells proliferate and blood vessels appear between them. Then glandular metamorphosis or transformation, when those cells they acquire the feature, they start to acquire the feature of glandular cells, and secretion the progesterone. Actually, in your textbooks there are no such stages. Just remember that. So yellow body, it exists during next 10 days, and in case pregnancy occurs, the chorionic gonadotropin of the implanted embryo produces chorionic gonadotropin, and it stimulates yellow body to stay and to develop further into yellow body of the pregnancy. But if pregnancy doesn't occur within 10 days, then yellow body degenerates, because the existence of yellow body is controlled by the luteinizing hormone, by insulin besides, and by chorionic gonadotropin. So chorionic gonadotropin produced by the implanted embryo is needed for the corpus luteum. In case of the absence of pregnancy, corpus luteum exists only 10 days. Then If without pregnancy it degenerates and is transformed into corpus albicans or white body. Here we can see those scar tissue replacing the yellow body, and every corpus albicans is the sign of menstrual cycle. So by counting the number of white scar tissue corpus albicans, we can count the number of menstrual cycles. Okay, and after we have discussed all structures, all follicles, all types of follicles present in the ovaries, we have to discuss the oogenesis as the maturation of oocytes. It differs significantly from spermatogenesis, despite the same phases it has. The first phase is the reproduction. Reproduction is when cells just increase in the number, they proliferate, and that's what occurs in embryonic period. So if in males reproduction continues along all the life, starting from the puberty, then in females only in embryonic period. Then the period of growth. Growth occurs just after the puberty, when they start to enlarge, and they are arrested, and before the puberty they just are arrested in the prophase of the first meiosis. And in this phase they all are frozen till the end of the life, till the end of the reproductive period. And then phase of maturation. Maturation occurs only in case of ovulation and fertilization. So only rare follicles, only rare oocytes can become mature, because maturation includes two meiotic divisions. All oocytes are arrested in the first meiosis, and they resume first meiosis only in case of ovulation, and they resume second meiosis only in case of fertilization. So predominantly primary oocytes, they do not become mature. Every menstrual cycle, every 28 days, only one oocyte ovulates, and only in case of pregnancy, in case of fertilization, secondary oocytes are transformed into mature ovum. So what you should remember is that all the oocytes are rested in the diplotenous stage of the meiotic prophase for 12 to 15 years. 12 because puberty starts at 12, and starting from this period some oocytes, they enter the menstrual cycle and resume the first meiosis, and up to 50 when they massively degenerate, so they disappear in general. Then what's written here is that maturation, it includes the first meiosis and second. And again, it is written that first meiosis is resumed only in case of ovulation, second meiosis only in case of fertilization. That's why, besides, we can say that blood Ovarian barrier is absent in humans, because there is nothing to hide from immune system. All oocytes are already familiar with immune system, as they exist in the organism since the birth. And secondary oocytes appear only once in 28 days. Mature ovum only in case of fertilization. And such a rare event is not the real stimulus, is not important for the immune system to be activated like that. So there is no blood Ovarian barrier. And here is the example from the USMLE database. The question is, if in brief, if a doctor decides to harvest the oocyte after its release from the ovary into the fallopian tube, in which stage oocyte will be arrested if it is not fertilized. Of course, you should choose the metaphase second. And you should not confuse with the prophase 1, because in the prophase 1, almost all follicles and almost all oocytes are there in the ovary. But if we are talking after the ovulation, if we are talking about oocyte just after its release from the ovary, after the ovulation, then such oocyte has resumed the first meiosis and is arrested in the second metaphase. Okay, just remember. Okay, and after we have considered the structure of the ovary, we go to the uterus. Uterus is a muscular organ to carry the pregnancy, and as I have said, it is destined to carry on one fetus. It is represented by such envelopes as endometrium, myometrium, perimetrium, and adipose tissue surrounding the cervix is also called parametrium, and it is put here for you not to confuse perimetrium with parametrium. Okay. So endometrium we are going to discuss in details because exactly here cyclic changes occurs along the menstrual cycle. Then as for the myometrium, it is represented by 3 layers and the borders between them are not very distinct as smooth muscle fibers they cross with each other, and we call such layers as stratum submucosum, vasculosum, and supravasculosum. The middle layer is called vasculosum because it contains large blood vessels, and myometrium contraction is controlled by oxytocin and is important during the parturition. Then, as for the endometrium, here the uterus is shown. If we are talking about the uterus, we should and as for the endometrium, we should say that inside the endometrium, 2 layers are considered, the functional layer and the basal layer. Functional layer is rejected during the menstrual cycle, and here we can see the endometrium, and functional layer is here, and basal layer is there. So functional layer is rejected every menstrual cycle, while basal layer is always preserved. This is the source of regeneration. Also, there is a difference in the blood supply. We can see that in the uterus, arteries, uterine arteries, they give rise to arcuate arteries, and they give rise to the radial arteries, from which 2 types of arteries arise. Side chains, they are called straight arteries, and they supply only stratum basalum, here straight arteries. And in the central axis, radial arteries, they give rise to spiral arteries, and spiral arteries, they nourish the functional layer of the endometrium, or the superficial layer. Why it is important? Because exactly those spiral arteries are sensitive for hormones and they undergo spasm if hormone concentration declines in the ovary. So now, after we have comprehended that there are 2 layers, the functional layer of endometrium and the basal layer of the endometrium, that they are supplied by different types of arteries, we have to discuss the cyclic changes in the uterus occurring along the menstrual cycle. So in the menstrual cycle, 3 phases are considered, and the first day of menstrual cycle is considered as the first day of bleeding, and in this way menstrual phase begins. What happens in the menstrual phase? First of all hormones, they decrease in the concentration, and we can see that especially progesterone, estrogens, luteinizing hormone are at the lowest concentration. Why? Because now yellow body degenerates in case of the absence of pregnancy. If there is no pregnancy, yellow body degenerates, and there is the decline in progesterone and estrogens in this way. While follicles are still immature, they do not grow, so there is no yellow body, there is no maturing follicle, so all hormones are at its lowest concentration. So the spiral arteries, which are hormone sensitive, if there are no hormones, spiral arteries, they spasm, and due to those contraction, they stop nourishing the functional layer, and the functional layer of endometrium degenerates, and we can see that it undergoes necrotic changes and is sloughing from the surface, it is lost. Then, due to this rejection, besides, if we return to the previous slide, we'll see that there are lots of veins in the function layer of endometrium, and they are exactly the source of bleeding, because arteries, they contract, so there is no arterial blood in the menstrual discharges, but there is the venous blood. It is dark in color, and this bleeding is not massive because it occurs through veins where blood pressure is relatively low. Then in the menstrual discharge, we can find the mucosa of the uterus, the endometrium with its connective tissue and remnants of dead tissues, tissue debris predominantly. And at the end of the menstrual cycle, the endometrium resembles the opened wound. As there is no epithelium here, the surface is naked, so it is recommended not to swim, not to bathe yet during this period, as endometrium is exposed and is very susceptible for different infections, as it is not covered with the epithelium. And then what happens after the menstrual phase? The next is proliferative phase, as during this phase a group of follicles start to grow. Why? Because in case of absence of estrogens, in case of absence of growing follicles in the menstrual phase, there is no estrogens, there is no estrogen in the blood, and by the mechanism of the negative feedback, we can see that follicular stimulating hormone increases in its concentration. You remember that estrogens are produced by granulosa cells together with inhibin. So inhibin inhibits follicular stimulating hormone. In case of granulosa cells absence, if they do not grow, if there is no growing follicles, nothing inhibits follicular stimulating hormone. It is uninhibited, and it increases in concentration, and it stimulates follicles to grow. As follicles start to grow, they produce estrogen. You remember, theca interna produces androgens, while granulosa converts them into estrogens. As estrogens increase in the concentration, here we can see growing follicles produce more and more estrogens, becoming more and more mature. Then in the endometrium, the epithelium proliferates and regenerates. That's why the phase is called proliferative phase. Proliferative because under the action of estrogens produced by growing follicles, under the action of FSH, this endometrium is re-established. If we return the previous slide, we'll find out that in the endometrium there are glands, and bottoms of those glands are hidden in the basal layer, while glands themselves are also present in the stratum, in the functional layer. So when functional layer is rejected, when it is degenerated, bottoms of glands remain in the basal layer, and they are used as the source of regeneration. Exactly out of here, epithelium migrates to re-establish the epithelial covering of the endometrium. So next phase and the last phase is the secretory phase, and here it is. And it is important to say what is there between the proliferative and secretory phase. Between those two events, critical process, critical event occurs. This is the ovulation. So as a result of proliferation, a group of follicles, they start to grow, and one follicle eventually ovulates. And starting from this period, all other follicles, they undergo atresia, while this follicle transforms into the yellow body. So during the next phase, secretory phase, we can find in the ovary the yellow body, but no growing follicles, because follicles, they undergo atresia. And yellow body continues to start to produce predominantly progesterone. So proliferative phase is also called estrogen phase, and secretory phase sometimes is called progesterone phase, because here this is the phase of the yellow body and the phase of progesterone. And it is very easy to remember, because in the secretory phase, yellow body appears, and by producing progesterone, it prepares uterus for implantation. Because just after the ovulation, fertilized oocyte travels along the fallopian tube to the uterus. And after 5 days, it should appear in the uterus and be implanted. So during these 5 days of oocyte traveling, uterus should be prepared for the pregnancy, and it is prepared by the progesterone, which is produced by the yellow body. So the main function of yellow body is to stimulate uterus to prepare for pregnancy. And now there are two different variants in case of pregnancy occurs. If pregnancy occurs, then implanted embryo starts to produce chorionic gonadotropin, and it helps to preserve the yellow body, and yellow body exists till the end of the pregnancy. But in case pregnancy doesn't occur, then there is no chorionic gonadotropin because there is no embryo implanted, and then yellow body degenerates within 10 days. And in case it degenerates, we start from the menstrual phase. Menstrual phase begins because in the absence of yellow body, we can see decline in progesterone and estrogens. As there is no growing follicles, no yellow body, then spiral arteries, they start to contract as they do not get hormones enough to perform their function. Okay, so those cyclic events, they restart every menstrual cycle. Every menstrual cycle, one of ovaries work, left or right, and as a result, we can see those cyclic changes. It is important to understand because, for example, different drugs which are used as hormonal contraceptives, what they do, they block they mimic the estrogen and progesterone, and in this way they block gonadotropin release and hormone synthesis. And if gonadotropin release and hormone is not produced, then the effect of those hormones are blocked. Ovulation doesn't occur, so luteinizing hormone is not produced. Ovulation doesn't occur, and as a result, yellow body is not formed, progesterone isn't produced, and uterus is not prepared for the pregnancy. So oral contraceptives, they block the ovulation, so there is no oocyte to be fertilized, and it prevents uterus from preparation for the pregnancy. So uterus, even in case of fertilization, uterus is not ready to accept this pregnancy, so it would not be implanted. Moreover, the cervical fluid is more dense and impermeable for spermatozoa and many other events. So that's how you can understand how different drugs work. Also, different hormonal pharmacological drugs are used, for example, to terminate the pregnancy, undesirable pregnancies or pregnancies with pathologies. On early terms, different progesterone receptors are blocked, and by blocking progesterone receptors, pregnancy cannot be prolonged. It is terminated preliminary because progesterone is needed to maintain the pregnancy. By blocking progesterone receptors, pregnancy is terminated on early terms. It is used. Also, some tumors, benign malignant tumors, are also treated by blocking receptors of progesterone or estrogens. And to understand how those drugs work, you have to understand normal functions of different hormones. So estrogens are needed to regenerate the epithelial covering of the endometrium, while progesterone is needed to stimulate secretion and to prepare uterus for the implantation. Okay, here those events are shown one more time, and as you can see, these are the cyclic changes in the ovary, these are the cyclic changes in the endometrium, and the respective concentration of hormones. So menstrual phase starts from the bleeding, and the first day is very exact. Then by the end of bleeding, we consider the beginning of the proliferative phase, and between proliferative and secretory phase there is the ovulation, also the very exact bodem. And then everything's quite clear. And here the image of uterine glands during the different phases. In the secretory phase, glands, they become much more coiled and they start to produce the viscous secret, lubricating the uterus and stimulating the implantation. And during next lecture I'll explain how placenta is formed in case of fertilization. In case of pregnancy, in the endometrium, placenta is formed. And also during pregnancy, cyclic changes occur within the mammary gland, and that's what we are going to consider next time. Now, menstrual cycle, and after we have considered the uterus, fallopian tube also should be considered. In the fallopian tube, there are three layers. The mucosa muscularis and serosa. Notice there is no submucosa. In the mucosa, two groups of cells are present, ciliated and secretory. Ciliated and secretory cells. Like in the respiratory epithelium, so it is often confused with trachea. As ciliated cells are very important, their sweeping motion help to transport the oocyte to the uterus, either fertilized or not. So oocyte is devoid of any organelles for movement. It doesn't have neither tail nor cilia itself, so it is passively moved by fallopian tube cilia. Another factor helping to transport the oocyte is the peristaltic contraction of the muscularis. And it is very important to understand, because if after the fertilization, during next 5 days, some drugs are taken by the woman, relaxing the smooth muscles, for example, to relieve the pain of any nature. Women, they sometimes take pills relaxing smooth muscle cells, and such drugs might affect the peristaltic contraction of the uterine tubes, and as a result oocyte is not properly transported, and after 5 days it occurs not in the uterus, but it is still in the fallopian tube, and it could be resulted in the ectopic pregnancy. So after 5 days, oocyte, fertilized oocyte, should be in the uterus and should be implanted there, but in case it is still in the fallopian tube, it could be implanted there, and ectopic tubal pregnancies are very dangerous and should be surgically removed, as fallopian tube is not the proper site to carry the pregnancy. Okay, so that's what you have to understand, that ciliated cells are important in transporting the oocyte, and muscularis, peristaltic contraction. These are the two factors helping to transport the oocyte. And female infertility is often associated with fallopian tubes because they could be blocked due to infectious diseases or different other problems, maybe just after surgical interventions. They could be replaced with the scar connective tissues. And sometimes if fallopian tubes are damaged, irreversibly damaged, then in vitro fertilization is used to assist in reproductive function, because you know that the fertilization occurs in the ampulla region of the fallopian tube. So in the fallopian tube there is the infundibulum, ampulla, isthmus, and intrauterine part, intramural part. And fertilization occurs in the largest, in the ampulla region. And if fallopian tubes are absent or blocked, then in the uterus fertilization is impossible. And that's why assisted reproductive technologies should be used to fertilize the oocyte in vitro, and then this fertilized egg could be implanted in the uterus. Okay, and at the end we should discuss the embryonic development of the uterus and fallopian tubes. And what should we say is that there is the same the same structure of reproductive system up to the 4th week of development. But starting from the 4th week, in case of Y chromosome is present, there is the SRY gene. SRY gene stimulates the synthesis of the testis-determining factor, which assists in the formation of the male reproductive system. In case Y chromosome is absent, in case SRY gene is not activated, then female reproductive system is formed. So, mesenchyma of urogenital ridges gives rise to theca interna cells, as well as in male reproductive system, Leydig cells derived from this source. Then, mesodermal epithelium covering those gonads give rise to primary sex cords and follicular or granulosa cells. So the same epithelium, by invading inside the gonads, gave rise to Sertoli cells in the male reproductive system. So Sertoli cells are homologs of follicle cells, and Leydig cells are homologs of theca interna cells. Then paramesonephric or Mullerian ducts, they give rise to the uterine tube, uterus, and cervix in the upper part of the vagina. So here the mesonephral ducts, they degenerate, and in case of the female system, some remnants are present close to the ovary, like epiophoron, paraophoron. These are the remnants of mesonephral Wolffian duct, because males prefer Wolffian ducts, while females, they prefer Mullerian ducts or paramesonephral ducts. So Mullerian ducts diffuse in the midline with the formation of the uterus, and primordial germ cells, they migrate from the yolk sac, populating the primitive gonads. And what we should say also, it is not put, I forgot to put, that vagina is developed in the upper region from the Mullerian ducts, and in the lower region from the endoderm of the urogenital sinus. So it has the double region of its epithelium. Then let's go to the example from the USMLE database. Here the uterus is shown, and which of the following is the most likely etiology of this malformation? You may see that uterus is double-horned. So here the mesonephral ducts incompletely fused, and due to incomplete fusion of paramesonephral ducts, we can see that uterus, it doesn't have the single cavity, but instead it is split into two. And the correct answer is incomplete fusion of paramesonephric or Mullerian ducts. And sometimes it occurs, and you know, in some animals this is the normal condition, and for especially animals, if multiple litters are characteristic for pregnancy. In humans, only one fetus is carried in the uterus, but in cats, pigs, in many animals, normal pregnancy occurs with several fetuses. In such animals, they have uterus with two cavities. In humans, it is not normal. Okay, I think another question should be put here. I forgot to pay attention last lecture, but let's now discuss this. A 16-year-old girl comes to clinic and she says that she has never menstruated. Physical examination reveal pelvic ultrasound development of ovaries. The genetic analysis here I want to highlight XY chromosomes. So 16-year-old girl, but genetic analysis XY chromosomes, and also genetic analysis it identifies nonsense mutation of a specific gene. Which of the following normal process in the sexual development of male fetuses is inhibited due to mutation of this gene? What does it mean, the mutation of this gene? Here, SRY gene is implied. SRY gene is located in the Y chromosome, and it orchestrates the male reproductive system development. So in case it doesn't work, female system is formed incompletely, of course, immature and improperly. but that's what I told you is very important in making the choice between the male and female. In case Y chromosome present with SRY gene encoding test as determining factor, then male reproductive system is formed. Sertoli cells appear, they inhibit Müllerian ducts and they orchestrate male system. But in case SRY gene doesn't work or Y chromosome is absent, then Müllerian ducts develop and female system is formed. Okay, in this slide we complete our first part of female reproductive system. Next lecture will be dedicated to the structure of placenta and cyclic changes in different organs and changes associated with pregnancy. So thank you for the attention. Subscribe to get the most recent information from our department. Goodbye.